So I'll start off by introducing and making sure I get the pronunciation of Kai Voss-Fels correct,
that was a good one.
rather than Voss-Fels, as is the common mistake. So Voss-Fels, who completed his PhD work in 2016 in Germany at the University of Gießen, which was founded in 1607. So just to put it into perspective.
Didn't know that.
And it's just north of Frankfurt, if people are familiar with the location of that. Where Kai, who is actually located in the animal sciences group in coffee, which has caused some degree of confusion with this seminar, actually did his work on wheat, barley, and rapeseed research projects. And he's going to be talking about some of the wheat work, where he's focused on quantitative genetics and looking at long-term genetic improvement. But his PhD work looked at resistance, breeding for biotic and abiotic stresses, and looked at genetic factors underlying root growth, and resistances against fungal diseases in wheat and barley. Kai started in coffee October 2017, just ahead of myself, where he joined Ben Hayes' group. And it seems that he's treated with some degree of suspicion there, where his animal science colleague thinks he's some sort of spy, given his background in crops and plants and so forth. But anyway, Kai is helping Ben with wheat and sugarcane breeding projects that Ben's actually involved in. And more recently, I've had the good fortune to have some chance to work with Kai on some theoretical work that we're looking at on extending quantitative genetic models to deal with non-additivity, epistasis, and genotype by environment interactions, and the applicability of that to some of the work that Ben and Kai are actually doing. But anyway, today, Kai is going to talk to us about a large-scale study investigating the impact of breeding on European winter wheat. So Ben Hayes has now turned up, and he missed my great introduction. But anyway, welcome, Ben, and I'll hand it over to you now, Kai.
Okay. Oh, wow. Thanks a lot, Mark, for this really nice introduction. I didn't know how old my former university is, actually. So, but now I know. It's pretty old. Yeah, it's a really great pleasure for me to give a talk today on some of the work that I've been involved in recently, and maybe a bit further info on why the heck would I talk about winter wheat breeding in Europe, here in Australia. Well, it's a pretty funny coincidence, I would say, that this day today, or exactly today 1 year ago, my wife and I were actually boarding the plane to move from Frankfurt in Germany to Australia. And yeah, literally the only things I brought was one suitcase, so one piece of luggage, and a pretty cool data set. And that's the one I'm going to talk about today. And in the first initial discussions I had with Ben Hayes, who's now my supervisor here at Quaffy, it turned out that he got pretty excited about the data set, and he agreed to get involved in the data analysis, because at that stage we've only finished the data collection steps. And yeah, this turned out to be really nice circumstance. As we finally got together a manuscript, we finished all the analyses, we pulled together a really nice manuscript and submitted this a couple of months ago. But I have to say, so this paper is still under review, so it's not citable yet, but we're of course, like always, hoping for nice and positive and gentle reviewers, so that we might be able to publish this work hopefully by the end of the year. So in this, I like to refer to this study as some sort of the success story of more than 50 years of winter wheat breeding in Western Europe, actually. And well, one of the many advantages of working with wheat, and I can tell you there are really quite a few, is that whenever you present your work in events like this, you don't really have to talk much about the crop itself, because everybody agrees that wheat is really important for global food security, especially in the future. Some people even say that wheat is the most important cropland in the world. Well, most of the wheat researchers say that, of course. So we all agree that we really have to significantly increase yields to be able to feed the world in 2050. So that's all kind of common sense, and we all agree on that. So what I much rather like to refer to as an introduction is this graph over here. So here you see the development of winter wheat yields in northwestern Europe in the past 50 to 60 years or so. And while I was basically just downloading the data from FAO Stat and plotted this. So you see on the x-axis you see the production year, and on the y-axis you see grain yield in decitons per hectare. That's the typical measure that Europeans use. So this is somewhat ranging between 3 to 8 tons per hectare, which is quite impressive. And while I was plotting this data in this graph here, I thought maybe some of you, since we're in Australia here, might not really be familiar with the importance or the significance of northwestern Europe for wheat production, for global wheat production. So I thought I might just download the data for Australia as well and plot it in the same plot. And this actually looks like this. So this is not at all meant to, make some sort of competition between growing regions or anything like that, because it's really comparing apples with oranges or so. In Europe, in northwestern Europe, we mostly grow winter wheats, which are growing for 2 to 3 months longer than spring wheats, which are predominantly grown here in Australia. But I guess this is just to highlight really the global significance of the production region. So in total, we're producing around 120 million tons per hectare compared to Australia, who's still, I think, ranked on rank 7 amongst the most important countries producing wheat in the world. But this region, northwestern Europe, is actually really, really important for global wheat supply. So there's at least 2 pretty obvious things that we can see in this graph here. First of all, we can see that yields have increased really dramatically, more than 2.5-fold if you compare the most recent production years with the first years where the records actually started. And secondly, we see that yields tend to be plateauing towards the end, towards recent production years. And that's actually kind of alarming, and many people are saying that we're really running into trouble with improving rates of gain per year to an extent that we will be able to feed the world in 20 or 30 years or so. So we took this situation here as a basis for the study that we were running, and we came up with 4 major questions that we were asking in this context. So first of all, we were really interested in the question: how high is the actual rate of genetic gain? Because you could always argue that these massive yield increases were mainly due to improvements of management practices, for example, that farmers simply have better or more chemical fertilizers, chemical plant protection at hand, for example. So we were really interested in the question: how genetically superior are recent lines compared to the old ones? Then secondly, and that's probably one of the most interesting questions for a geneticist, which genome regions are actually important. Because if we knew about the genetic regions that are underlying breeding progress, we could probably use that information to even further improve future cultivars. Then one really hot topic: did breeding actually impact genetic diversity? So it's a really widespread belief that strong selective breeding for improved yields under high input environments actually narrowed down genetic diversity to an extent that we don't have enough diversity left to make further gains in the future. So we try to address that question as well. And last but not least, the by far the most difficult question to answer: how far can we actually go? And I'm not going to make a claim that I can answer this question. I don't think anybody can, really. But I'm going to show you some estimates that we obtained based on the data set we had at hand, which is quite interesting still. So let's get started off with a statement that I really like, saying modern wheat varieties only perform well under high chemical inputs. I mean, this is a statement that really most of the people outside the field of agronomy or agricultural sciences would absolutely agree with. So I'm pretty confident if I just went out on campus and asked 100 people, I would say the majority of people outside ag science would agree with this statement. But of course, as geneticists or breeding researchers, we weren't really convinced, and we were asking the question: how high is the actual rate of genetic improvement, if there is any? So that was like a first major question that we wanted to address. So must have been in the beginning of 2014 when we started a pretty large collaborative project between German research institutions at first. And we started off by sampling around 200 high-yielding European elite cultivars from the last 6 decades of winter wheat breeding or winter wheat production. And it's really important to consider here that this is not a classical diversity panel or so that got randomly sampled, from across the globe or randomly sampled across a production region. But these lines are what we believe to be the best of the best from the last 50 to 60 years of wheat breeding and wheat production in terms of agronomic and economic importance. So that's really important to consider here. So then we were running really extensive field trials in Germany across 5 locations in 2 years. We were actually running the field trials across more years, but the data that went into our analysis comes from 2 years. And the coolest bit about this project really is that we not only tested those lines under very high chemical inputs with chemical fertilizers and plant protection, but we actually applied 3 different management intensity levels, ranging from very high input, where we applied full amounts of fertilizers, mostly nitrogen obviously, and plant protection, chemical plant protection, mainly fungicides, because that's the biggest problem in Germany at least. Then we grew them in the same trials in a medium input environment where we did apply fertilizers, but no chemical plant protection at all. So this is really interesting because here the plants, they do have enough nutrients, but a quite significant biotic stress component really kicks in this intensity level. And we also grew them in very low input environments where we didn't apply any fertilizers or any plant protection at all. So we ended up with 30 kind of unique environments in which we measured a whole array of traits, including grain yield and the most important grain yield components, different qualities, quality relevant traits, resistances. So we basically covered pretty much all important fields that plant breeders are working on. And as for the genomic data, we had SNP array data at hand from the 15K Illumina SNP array. And here I have to mention that with input or with help from Matt Hayden based at AgriBio, we were able to get actual physical positions of all the markers because they have quite amazing genomic data sets. So that was really helpful, or turned out to be really helpful for our subsequent genetic analyses. So that's the data set. And maybe let's jump into one of the most interesting questions when we talk about genetic gain. How does grain yield look like in those different varieties, in those different intensity levels? So here on the left hand, you see yield in decitons per hectare plotted against the year of registration of the respective cultivars that we tested in our panel. And we see a really nice, pretty much linear association between the year of registration and grain yield. And if we're talking about genetic gain and compare the oldest versus the latest 20% of the lines, we see improvements of more than 16%, which is quite impressive, I would say. However, it becomes way more interesting if we look at the medium intensity level here in the middle, where we did apply fertilizers, chemical fertilizers, but no plant protection whatsoever. And here, the newest 20% of the lines were actually more than 30% better than the old ones, which is really impressive and really amazed us a lot. And we saw a similar trend even in the management intensity level, where we didn't apply any chemical fertilizers or chemical plant protection. So here again, almost 27% yield increase of the latest versus the oldest 20% of the varieties. And as it turned out to be it was quite a task, because we had so much traits and so much information. So we were thinking of how can we actually represent this in the most comprehensive way. So we came up with a way of graphically representing our work in the last couple of years by something like this. So here you see grain yield, and what you see here is that each of those lines represent basically one cultivar tested, so one of the 191 cultivars tested. So each cultivar has 3 data points, grain yield in high, medium, or low, input management system. And the colors, they refer to the year of registration where the respective variety was grown in. So the very, very bright yellow ones are the recent varieties, whereas the blue ones are the old ones. And we kind of normalize the data, because obviously you can't compare absolute measurements of all the traits. So we normalize the data ranging from 0 to 1, where 0 is the worst and 1 is the best in terms of phenotypic measurement. And as I was saying, we didn't only do this for one trait, but we actually did this for 17 traits measured in all different in all different field trials across all intensity levels. So we ended up with I find a pretty cool graph like this that's basically summarizing a couple of years and I don't know how many hundreds of thousands of euro of phenotyping into one really comprehensive graph. And I have to say as well that the publication also features additional data from a single trial where we actually did have severe droughts in the field. You know, you could imagine probably that it's not really straightforward to measure drought in the field in Germany. Well, it turns out to become more and more of a problem in recent years. But yeah, we were lucky enough to have at least one field trial where we did have really severe droughts, and the trends are pretty convincing. So that's just a kind of a teaser. So once the publication comes out, I mean, if you're really interested in drought tolerance, so this is also featured here. I mean, about this, you could probably talk for a few hours about this figure alone, but there's like a few things that are really interesting. For example, if you look at plant height, well, it's pretty common sense that old varieties tend to be pretty long, and then breeders selected for shorter and shorter lines in order to increase the cultivar's capacity of carrying more or increased grain loads. But this only went to some extent. So at some point, breeders actually bred backwards to increase lines again, and that's pretty interesting to see, because the most recent lines are actually clustered here somewhere in the lower third. So the most recent lines aren't the shortest, but they are somewhere clustered in the lower third here. So bottom line of this work, of this phenotypic work here, is that modern varieties, they perform way better under optimum or high input systems, but they do perform extremely well, especially under abiotic and biotic stresses. And that's a really, really important statement, we believe. Also like a really important political statement. Especially in times where organic farming is becoming a thing in the really rich and wealthy areas of the world. So we hypothesize based on our data set that the most recent varieties that have been bred using the latest technologies are actually the most suitable cultivars, also to be grown under organic farming systems. But what about end-use quality? So wheat is really not at all only about increasing grain yield. So there are really, really strict requirements in terms of quality characteristics, especially for end-use quality in terms of baking quality. And we also covered that. So amongst those 191 varieties that we were testing, we basically covered all important quality classes we have in Germany. So there are 4 major quality classes, starting in the very left. The E-wheats, they have an extremely high protein content, are typically only used for export. Then we have A-wheats, which have a very high protein content and are typically used in milling to offset varieties with lower protein contents. We have B-wheats, the classical baking wheats, which have decent contents of protein and are ideal for baking. And then we have C-wheats, which have typically very low protein contents, and they are used in animal breeding. And it's pretty interesting because most of the breeding aims to increase protein content, because this is where the farmers make the most profit of, like selling high quality wheats. And I think only 1/3, or not even 1/3 of the wheats are actually used for baking. Whereas 2/3 or so are used in animal production, which is pretty interesting. So if we look at different quality parameters, we have a situation like this. So up here you see crude protein, so that's basically protein content of the seed. In the 4 main quality classes, I blanked out the C quality class because that's not so important for baking quality, at least. And it's pretty interesting because we see a pretty significant decrease in crude protein which was pretty surprising. But if we look at other quality parameters, such as sedimentation value, for example, which is basically saying how well suited the grain or the flour made from the grains is for baking in terms of volume, so the higher the better. So we see a stable to a slight increasing trend, which is very interesting. If we look at falling number, and again, this is a quality characteristic where the higher is the better, we see quite significant increases in falling numbers of recent varieties. And in the end, we see very significant increases in grain yield. So, and then that's really interesting, I would say, because in summary, this means that modern varieties, they have a lower crude protein content, so lower content of raw proteins, but it looks like major quality parameters, they stay stable or get slightly improved even, while we also have highly increased grain yields. So the bottom line would be that we see a huge progress actually in quality component optimization, most likely due to breeding selection, which is really interesting. So coming to the next major question, which genome regions are important? And as I was saying, that's probably one of the most interesting questions for a geneticist, because if we knew the regions, we could probably explore them in breeding, in modern breeding, to improve future cultivars. So we thought, well, we could probably use a classical kind of QTL mapping approach or genome-wide association mapping approach, but since our sample size was still quite small, I mean, we're still dealing with only about 200 individuals, and we thought we might not have the best statistical power to actually detect genetic variance. So we thought it might be worthwhile to use the genomic prediction framework to identify chromosomal blocks that have a high variance in terms of trait variance. So what we did here was we basically calculated genomic estimated breeding values, not for single markers alone, but we grouped markers based on linkage disequilibrium. So we basically built haplotype blocks, and we summed up all predicted marker effects in these blocks. And then in the end, we basically calculated variances across the different haplotype effects. And so this is getting a bit more technical to guide you through the algorithms or the method that we were actually using. So you can consider a chromosome that is 50 centimorgan long and has 30 markers, kind of evenly spread across the chromosome. So what we did in the first step was we predicted marker effects for each of those single markers using the ridge regression blood framework that was introduced by Ben Hayes and colleagues in 2001. And then, but I mean, one typical feature of this method is that each marker does get an effect, and this effect is typically pretty small. So it's really hard to tell based on this or based on these effects which marker is actually important. So we included another step in which we defined haplotype blocks, and there's at least 3 different ways of doing that. So you could either group markers based on their genetic or physical distance. You could, for example, come up with blocks of 5 centimorgan, and you would just basically slide across the chromosome and always group markers that are in 5 centimorgan windows. You could also do it in theory based on marker number, where you define a block of markers as always as 3. But with wheat, we're running really into a problem that using those 2 approaches, you might end up grouping markers together that are literally light years apart in terms of distance on the chromosome. So they didn't seem to be very optimal in the case of wheat. And so we actually used the third of the suggested options here. So we grouped markers based on linkage disequilibrium. So you could linkage disequilibrium, that's basically the non-random association of alleles at different loci. So you basically take into account that for some combinations of alleles, there's not much variation, so they tend to be inherited together. So that's why you just group them together in one block. You can do this based on a threshold, so that's how we did it. And once you've done that, you can actually, so you would come up with different haplotypes for the different blocks. So for example, this big block over here, this might have 6 different haplotypes or alleles. And each of those haplotypes gets assigned an effect that was obtained by just summing up the effects of the markers in this block. So we do this for all the blocks on the chromosome or on the whole genome in the end. And then we can calculate variances across or among those haplotype effects. And then in the very end, we can instead of plotting single marker effects against their genetic positions, we can actually plot plot trait variances of the defined LD blocks against the position of the block in the genome, which is actually turned out to be a really nice way of dealing with a small population. So looking at some actual data, here you see, as an example, chromosome 1A from wheat, and so here I've used RR-BLUP to predict marker effects of all the markers that were mapped to chromosome 1A. So the trait here was grain yield under high input, under high chemical inputs, and as you see, the range of the marker effects is typically really, really small. So considering that you have varieties in that panel that can easily exceed 10,000 kilos per hectare, we're talking about between -2 to +2 kilos per hectare. So it's really, really hard to tell which one of those markers is actually important or which one has a higher effect than the other. So we can basically summarize, we have a lot of very, very small effects here. But at the same time, we know that for wheat, and also for most of the other crops that we use and eat, we have very, very strong and pronounced linkage disequilibrium structures across the genome, as indicated here by this LD heat map. So we know that single markers aren't independently inherited, or their alleles, but we know that they are jointly inherited in blocks. For example, these markers here, they form a pretty significant and tight linkage disequilibrium block. So we came up with an algorithm, basically, in which we firstly generated a genome-wide haplotype map. And turned out that one of my colleagues, Elizabeth Ross, was really helpful with setting this up in terms of scripting and programming. So we generated a haplotype map. Then we summed up allele effects within those blocks as described earlier. And then we identified blocks with high variances for all the different traits that we were looking at. So talking about grouping markers based on linkage disequilibrium, that's kind of a summary figure that shows the situation across the genome. So on the x-axis you see the 3 different subgenomes of wheat, A, B, and D, with 7 chromosomes each. And on the y-axis you see the sizes of the blocks, of the linkage disequilibrium blocks, in terms of a number of SNPs that they contain. And it's pretty interesting. So the initial, say, 8,700 polymorphic SNPs were grouped into 3,700 linkage disequilibrium blocks based on the threshold of 0.7. So we see the by far biggest block is located on chromosome 2D up here, but all the other big blocks, they're kind of evenly spread across the genome, which is interesting. And if we look at distributions of differently sized blocks across the genome, we see that around 2/3 of the blocks only consist of a single marker. This has most likely also something to do with low marker coverage. Around 13% of the blocks include exactly 2 markers, but around 22% of the blocks have more than 2 markers actually. So if we then plot the variances of those blocks that we defined based on LD against their position in the genome, we can create figures like this here. So this is just showing subgenome A as an example, and each of the slices here of this pie chart basically represent one single chromosome. And the trait we were looking at was grain yield. So the inner circle you see here, that's just representing the sizes of the blocks that we've defined in terms of number of SNPs that they contain. And then those 3 outer circles here, they represent the variances that we estimated in each of the 3 intensity levels for grain yield. And I want to I want to maybe just highlight two interesting blocks that we found here. So we find some blocks, like the one on chromosome 1A, that have somewhat of a high variance across all three management intensity levels. So there must be some common genetic variant underlying trait expression here, whereas we also have blocks that have intensity-specific variances, like the one on chromosome 5A. So when we estimate variances under high input environments for grain yield, we don't see a high variance here, but this thing is popping up in the medium and in the low input environment, which probably indicates that there's some sort of resistance gene or a resistance factor underlying. So that's really nice to see. If we look at correlations of the, estimated haplotype effects, we also see some pretty interesting trends. So first of all, we calculated genetic correlations between high and medium or high and low input environments, and we found fairly high genetic correlations between those measurements, indicating that there's quite a big, like an overlapping basis, genetic basis for trait expression of grain yield, or for grain yield in three different intensity levels. So we can, and we can actually plot those effects as well in a, oh, it's not working. Doesn't matter. So we can also plot the effects, the estimated haplotype effects in a 3D plot. And so this thing would normally rotate, and it would nicely show that those haplotypes that have a very strong effect under high input environment tend to also have a pretty high effect under stress environments, which is really interesting. And when we look at, sorry, just 1 second, having a problem with the remote.
Killed the computer.
No, not really. It's weird because it's changing the slides on the computer but not on the screen. It's not doing. Yeah. Yeah. Let me check.
Is it not? There you go.
Oh, beautiful, thank you. Yeah, so if we consider the top 100 linkage disequilibrium blocks with the highest variances in each of the three intensity levels for grain yield, and we look at how many of those actually overlap, how many of those are actually the same, we find a fairly high number of blocks that are actually affecting yield in all three environment levels. Well, there's some that only have a high variance under high input, some that have a kind of intensity level specific variance under medium input, and some that have a high variance under low input. But the bottom line is that it looks like that kind of similar genes are actually involved in the trait expression, grain yield, in this example, under optimum end stress. And that's actually really important, because this would mean that improving grain yields under high input systems also improves grain yield under stress environments, which is actually pretty good news for breeders. So we also looked at the number of detrimental haplotypes in the course of breeding, and we defined detrimental haplotypes as haplotypes that have a zero effect on yield or even a negative effect on yield. So here, this graph I showed you earlier, so here you see the developments of yields depending on the year of variety release. But if we then plot the number of detrimental haplotypes for each of those lines against the yield increase plots, we see pretty interesting and pretty striking trends. So we were considering the 100 blocks with the highest variance, and we were basically just counting in each of those lines how many haplotypes with a zero or a negative effects are these carrying. And it's a pretty, I mean, these graphs are pretty striking, almost mirror images of the development of yield. And if we zoom into outliers in each of the 3 intensity levels that have an outstandingly low performance, we would also find that those lines are the exact same ones that have an outstanding high number of detrimental haplotypes. So in summary, based on these findings, we can say that breeders have actually purged detrimental alleles from modern germplasm. Well, not completely, but to an extent that in modern varieties, they actually do contain a significantly lower number of bad alleles than old ones. Next important question we wanted to address is, did genetic parameters change over time? Did breeding actually really reduce genetic diversity? So first of all, we looked at population structure amongst the lines that we were testing in our study. So we calculated genetic distances among those lines, and we plotted population structure here in a principal component analysis plot. And to make, yeah, well, we also color coded the oldest and the newest 20% of the lines in different colors. And what we effectively see is there is no population structure. But this kind of really makes sense, because this reflects the fact that breeders in Europe have really extensively been exchanging their material. So in Europe, if there's a really successful variety, it's very likely that other breeders will use this variety to make crosses themselves. So breeders obviously have been doing this a lot, to an extent that pretty much all the genetic material that we're looking at is pretty much the same in terms of population structure. We then also looked at very popular population genetic parameters, probably some of the most important ones or most popular ones: linkage disequilibrium and gene diversity, or expected heterozygosity. And we calculated those measures as means across subgenomes, but not in sliding windows across the genome, but rather in sliding windows across genotypes. So we would group the 40 oldest genotypes here, calculate mean R-square and gene diversity measures, then slide the window one genotype to the right, do the same thing again. So we ended up with 150 or so measurements, or mean values in the end. And we got pretty interesting trend curves that you can see over here. So here you see expected heterozygosity or gene diversity. Those are the two same things. And they look pretty busy, and you see a lot of fluctuation going on depending on the year of registration. So down here on the x-axis, you see the mean year of those 40 genotypes that were located in each of those sliding windows. And on the y-axis, you see gene diversity. And if you compare the oldest versus the newest varieties, it's pretty interesting to see that, for example, in the A subgenome, we actually see a pretty striking increase in gene diversity of modern lines compared to the old ones. For some reason, we see a decrease in the B subgenome, but for the D subgenome, again, we see an increase. So we can't really see a really clear pattern that gene diversity was really significantly reduced across the genomes. So we also looked at linkage disequilibrium as a really popular measurement, and we found similar trends. So LD actually decreased in the A subgenome if you compare the oldest with the newest varieties. It decreased for the D subgenome as well. It looks like it's slightly increased for the D subgenome, even though the trend doesn't really look extremely alarming or extremely striking. We also looked at single chromosomes, and this is just an example over here. This is chromosome 5b, and if we plot LD measurements in sliding windows against the year of registration, we see those interesting peaks on single chromosomes like the one here, and this kind of makes sense if you think about it. This could really represent the fact that there were some really successful varieties in the past that after being released have been used by pretty much all the breeders. For introgression, for example, they carried a really important resistance on chromosome 5b, so all the breeders were using them. And then you would, and this would make sense in terms of you would expect LD to increase significantly here, but then as a consequence of recombination events and so on, this LD peak relaxes again in the course of breeding. But these patterns are actually pretty interesting to see. So in summary, we can't really say that we see a strong genome-wide trends for any of those two important population genetic parameters. So we can't really see a significant reduction in genetic diversity in our population that we looked at. And this really nicely aligns with a statement that Hans Braun made earlier this year in a big wheat meeting. So Hans Braun is leading the global wheat breeding program of CIMMYT. And he said they can't really find evidence in their germplasm pools that genetic diversity is actually being reduced. So that's pretty interesting because this really aligns with what we found in our data set. So we've almost made it through. Last big topic that I wanted to cover, how far can we go? Well, I told you earlier that's really hard to answer, but we made some attempts to get a feel for what's actually there in terms of capacity in the data set that we were looking at. So what we did was for grain yield under high input intensity, we considered the 20 highest yielding varieties, the 20 best cultivars with the highest yields, and plotted them down here. So they were ranging somewhere between 8.5 and 9.5 tons per hectare, which is really a lot already. And then we considered different scenarios in which we thought what would actually happen if we, for example, replaced bad haplotypes for really important LD blocks with the best possible haplotypes. So we considered a scenario where we replaced the bad alleles with the best alleles for the top 20 haplotypes. And this resulted in a potential yield increase of a bit more than 1%, which is really not too bad if you think about that we're currently probably only generating around 1% yield increase per year, maybe even less. So that's actually not too bad. Considering the top 50 blocks, we could get estimates of yield increase of around 2.6%, which is really not too bad and quite significant. Increasing this number up to 100, we can almost generate 5% yield increases. But here, if you want to consider 100 loci spread across the genome, the situation is getting a bit tricky already. But then in the last scenario, we thought, okay, when if there were no limitations in terms of stacking the best possible variance up, we were considering a scenario where we would stack up the best possible haplotypes across all 3,700 blocks. It was quite impressive to see that this would potentially yield in 25% yield increase, obviously making very simple assumptions, ignoring all sorts of interactions between loci, just assuming full additivity. But this really, and it's not really realistic to stack all genome-wide haplotypes up, but I think that's just really nicely highlighting how much of sleeping useful variation there is, even in a very narrow elite germplasm pool like the one of winter wheat in northwestern Europe. So to summarize, we see really massive genetic improvements of modern varieties, and most importantly, we find them not only under high input optimum growing intensities, but also, or particularly under stress environments where we don't apply any fertilizers or any plant protection. And that's an extremely important statement. This really says plant breeding has been extremely successful in improving the genetic capacity of modern varieties to deal with really challenging environmental conditions. We kind of came up with a new kind of LD-based haplotype approach that uses linkage disequilibrium as a basis to obtain or to identify blocks in the genome that have a high effect on the trait of interest. And we do think that this might be a really suitable approach for especially dealing with small data sets, a situation that most of the crop plant researchers are familiar with. At the same time, we can really nicely show in our data set that breeding actually removed detrimental alleles over time, and we can't really see any trackable effects on important population genetic parameters like linkage disequilibrium or genetic diversity. So it's probably more like finding the most optimal combination of favorable haplotypes instead of actually eliminating genetic diversity. And yeah, last but not least, how far can we go? Well, theoretically, there's a very high potential, even in this very, very small data set that I'm showing here. But realistically, the gain that we could probably achieve is way smaller, but I'd say it's still considerable. If you just consider, say, 50 loci across the genome, so you can actually generate considerable gain. Of course, modern breeding tools like hold great promise. Genomic selection, most importantly, many people are working on this, and this is becoming actually really important for modern wheat breeding as well. Speed breeding, well, Lee Hickey, that you probably all know, he promised me that he can speed breed winter wheats in 1 year from now. So I guess I just wait and see how he goes. Genome editing. Everybody's talking about genome editing at the moment. It's maybe not so feasible for genome edit a whole number or a huge number of loci across the genome, but this might be something that we could keep in the back of our heads. So this might be an interesting tool as well. So to really sum up what I initially started with, I hope this study really convinced you that modern breeding actually secures sustainable wheat productivity. With this, I'm pretty much done. So the project where this data was generated in is called the Brevex project. There's also a nice website that you can find more information. I think also a bit of history about the wheat cultivars that we were using. There's a whole lot of partners involved. Most importantly, my former colleagues from Germany, Andreas and Benny in the group of Rod Snowden, that did a lot of the really hard work. My new colleagues here at Quorfie at the University of Queensland, Elizabeth Ross, most importantly, and my current boss, Ben Hayes, who gave really critical input for the data analysis here. I'd like to thank all the funding bodies, all the other partners as well, of course, and I really thank you for coming along, and thanks a lot for your interest and for your attendance. Thanks.